Hello everyone, my name is Vivek Modi and I am a postdoc in Professor Roland Dunbrack's lab at Fox Chase Cancer Center in Philadelphia. On this poster, I am going to present KinPhyCoRe, a web resource for protein kinase sequence, structure and phylogeny. This figure displays a typical structure of protein kinase with the ATP binding active site in the middle region of the protein. It contains the conserved DFG motif, which adopts a unique conformation in the catalytically active state and multiple different conformations in the inactive state. As an example of its conformational diversity, the figure on the right displays multiple conformations of DFG phenylalanine in the structures of EGFR kinase. In our previous work, we developed a scheme to cluster and label all the conformations of DFG motif. We first group all the structures into three categories based on the location of DFG phenylalanine in the binding pocket. These three spatial groups are labeled as DFG in, DFG out, and DFG inter for intermediate conformations. Within these groups, we cluster based on the backbone and side chain dihedrals of DFG motif residues. Each conformational label consists of a spatial group plus a dihedral cluster. According to this nomenclature, the ATP and magnesium ion bound active state of the kinase is labeled as DFG in BLA-, while the conformation in complex with type 2 ligands is called DFG out BPA-. I encourage the listeners to read our paper to understand the details of clustering and nomenclature. With this background in mind, in the current work, I introduce KinPhyCoRe. In the next few slides, I will show how a user can access information on kinase conformations, inhibitor classification, protein kinase sequence alignment, and phylogenetic tree. The users can access the website through this link, which will take them to the home page. The navigational buttons provide access to all the information in different ways. The simplest among these is Scan on Browse button. Clicking Browse will take the user to this page where all the protein kinase chains are listed in one table. The table contains identifiers like gene and protein names, UniProt and PDB IDs, and ligands bound in the structure. Each chain is labeled with a conformational assignment like DFG in BLA-, DFG in BLB+, etc. as described earlier. We have also classified the different types of ligands based on the region of binding pocket they bind to. The labels for ligand types are listed in the last column. Each blue element in this table is a hyperlink, which will take the user to specific pages. For example, if the user wants to see all the structures known for a particular gene, she can click on individual gene names here. The same concept is used for other columns also. On the gene page, user will find information about its phylogenetic group, domain boundary, etc. This page shows aurora kinase as an example. For a quick analysis, we list representative structures which are displayed using NGL Viewer on the right. The table at the bottom provides the list for all chains for that gene with additional information like mutations and activation loop residues, etc. On each page, we also provide buttons to easily download data. The database table itself could be downloaded as a tab-separated file using this button. Moreover, the structures for representative PDBs or all of them could be downloaded in PyMol sessions and coordinate files itself from these buttons. Like the browse page, the information on this page is also hyperlinked to other pages. For example, the user can click on a conformational label to get all the structures in that cluster. This page shows all the structures in DFG out conformation. The top table provides statistics of DFG out conformation across different kinase phylogenetic groups with a representative structure. As shown on the gene page, we provide download buttons for PyMol session and coordinate files, and all the structures can be downloaded with just one click. The table at the bottom lists all the structures in DFG out conformation with the annotations described before. PDB ID will take the user to individual PDB page. In addition to the annotation described before, we also provide the UniProt sequence for the protein with information on mutation, phosphorylation, and unresolved residues. The user can also click on ligand identifier to reach the ligand page. Here as an example, we see all the structures in complex with the ligand BB8, which is also called commonly bosutinib, an important drug in cancer treatment. This drug is found in complex with different conformational clusters, which is critical information in drug design. This example shows the utility of our clustering and organization of the database to gain new biological insight. Another important feature is the search page. If the user wants to retrieve any specific information, she can use our search page and query the database using unique identifiers like gene names or PDB IDs or a combination of kinase phylogenetic group, conformation or ligand type. We have also developed a web server. Another important feature. The user can upload their kinase structure file to determine its conformation. This could be very useful for kinase structures modeled by using a software or generated from a molecular dynamic simulation. In our previous work, we also developed a structure-based sequence alignment of all human protein kinases and used it to update kinase phylogenetic tree. The user can access and download this information from our web resource. We hope that KinPhyCoRe will be useful for research community and facilitate future work. If you have any questions or comments, then please feel free to contact us. Thank you very much.